Hello students, welcome back to my biology online classes. Today I am going to discuss with you about this topic, brief summary of three types of RNA. In my previous three lectures, the major three types of RNA, rRNA, mRNA and tRNA was discussed in detail. So now it's time to study the brief summary of these three types of RNA. There must be some characters. Based on those characters, the three types of RNA will be discussed. So students, let's concentrate about the brief summary of these three types of RNA. The first point about characters is weight. It's about weight of these three types of RNA. So write the first point, weight. When we are going to compare about the weight of these three types of RNA, rRNA is heaviest. mRNA is heavy. And tRNA is light in weight. So the first character suggests that among these three types of RNA, rRNA is heaviest and tRNA is lightest. The second point of difference, the next character of difference is point number 2, percentage of total RNA of the cell. The next character is about presence of percentage of these three types of RNA in cell. rRNA percentage present in the cell is about 80%. A large amount of rRNA is present. In case of mRNA, it is 3 to 5%. And in case of T, this is 10 to 20%. Okay. So, in case of presence of RNA, percentage of total RNA of the cell, as we can compare, rRNA is present in highest amount, almost 80%, then tRNA 10 to 20% and mRNA very less, 3 to 5%. Now concentrate about the third point of difference. The third character to be differentiated among these three types of RNA is sedimentation coefficient. Sedimentation coefficient. In my previous three lectures, particularly in discussed about, I had discussed about the rRNA. In that lecture, sedimentation coefficient, what is sedimentation coefficient was discussed in detail. So if you have any confusion about sedimentation coefficient, please consult the lecture about rRNA. Okay, 28S, 18S, 5.8S, 23S, 16S and 5S. Sedimentation coefficient in case of rRNA, and what will be in case of mRNA? This one is 8S, and in case of tRNA this one 3.8S. Now the next character to be differentiated, point number 4, number of nucleotides. Number of nucleotides. In case of rRNA and in case of this 5S RNA, 5S rRNA, the number of nucleotides present is 120 nucleotides. In case of 16 to 18S RNA, 16 to 18S rRNA, 1600 to 2500 nucleotides. I am going to use abbreviation MT. And in case of 23 to 28S RNA, 23 to 28S rRNA, the number of nucleotides are 3200 to 5500 nucleotides. About number of nucleotides present in these three types of RNA. rRNA have three categories. In case of 5S rRNA, it is 120 nucleotides are present. In case of 16 to 18S rRNA, 1600 to 2500, 1600 to 2500 nucleotides are present. And in case of 23 to 28S, 23 to 28S rRNA, 3200 to 5500 nucleotides are present. Okay. And in case of mRNA, 900, I am going to use a separate color, 900 to 1500 nucleotides are present, and this can be seen in case of bacteria E. coli. So mention E. coli here. Only 73 to 93 nucleotides are present. The next point, your next character is point number 5, molecular weight. Molecular weight. 23S rRNA, it will be 0.1 to 10 to the power 6. 30S rRNA, the molecular weight 0.55 into 10 to the power 6. So, in case of rRNA, while considering 23S rRNA, it is 1.1 into 10 to the power 6, and while considering 30S rRNA, it is 0.55 into 10 to the power 6. Here it will be 5 lakhs. It will be 25,000 to 30,000. So this is about molecular weight of these three types of RNA. Usual bases. Unusual bases. Small amount of methylated base is present. E. coli, it is 1 part 100 to 150 nucleotides. So the unusual base present in case of rRNA, it is small amount of methylated base is present, and it is observed in case of E. coli. What amount? In what amount it is present? 1 part 100 to 150 nucleotide, means one methylated base is present. If we consider one methylated base is present out of 100 to 150 nucleotides. Let's see the case in case of mRNA. Very small amount of unusual bases is present. Small amount of unusual bases are present. Okay. And in case of tRNA, content of unusual bases is present in case of tRNA. High content of unusual bases are present. And if you want to know about the detail about unusual bases which are present high content in case of tRNA, please go through my lecture about tRNA, and in that lecture these unusual bases and all the structure of tRNA was discussed in detail. Okay, so the next character is point number 7, character number 7. Here in case of E. coli, 1 per 35 to 40 nucleotides are present. Means in case of E. coli, out of these 35 to 40 nucleotides, only one unusual bases are present in case of E. coli. Okay, if we consider E. coli as an example. So the next character is site of synthesis. Site of synthesis. It is derived form nuclear DNA. Derived from nuclear DNA. And in case of mRNA, it is synthesized in nucleus on DNA template. It is also synthesized in nucleus like mRNA. It is also synthesized, right, same to same here in case of tRNA. It is also synthesized in nucleus on DNA template. Beginning of synthesis. Beginning of synthesis. Synthesis begins at gastrulation, increases as development proceeds. In case of mRNA, some mRNA is found in ovum. New mRNA synthesized during cleavage. And in case of tRNA, synthesis occurs at the end of cleavage ends. Synthesis occurs at the end of cleavage ends in case of tRNA. The term cleavage, gastrulation, it's all found in developmental biology. These terminologies are found. So the next point, next character is of relationship DNA. In case of rRNA, no obvious relationship to DNA because rRNA is formed from small sections of DNA. In case of mRNA, shows base relationship to DNA. base relationship form all section of DNA. Form all sections of DNA. If you want to study about this base relationship form all sections of DNA, you can consult my lecture about mRNA. And what's about tRNA? Same as in case of rRNA. Same as rRNA. Means no obvious base relationship with DNA. Next and last point is about function. Paired base in case of rRNA, unpaired base may bind mRNA and tRNA to ribosomes. Okay. And in case of mRNA, convey genetic information. Convey genetic information from DNA of chromosomes to the ribosomes where it takes part in protein synthesis. And what's about tRNA? It acts as adapter for attaching amino acids to mRNA template. So this is the brief summary of these three types of RNA.